Hello everyone. So today in this class we are going to discuss chemical carcinogenesis. So it involves three steps. First is initiation of carcinogenesis, promotion of carcinogenesis and progression of carcinogenesis. We'll be discussing in detail each step. So first is initiation of carcinogenesis. That means a particular chemical it will initiate the carcinogenesis when a person is exposed to any chemical for a short duration in a large dose or for a longer duration. So then this initiation will be done. So the change that produced during this carcinogenesis initiation is very sudden and it's irreversible and is permanent. Okay. So here again these carcinogens were again of two types, that is direct acting carcinogens and indirect acting carcinogens. So the name itself indicates the direct acting carcinogens. That means the chemical directly induces carcinogenesis without undergoing any metabolic process. Okay. And indirect acting carcinogens or pro-carcinogens. Pro means they were not active carcinogens. So when they undergo some metabolic reaction, whatever the metabolite that is coming out, so that will act as a carcinogen. So it is also called as indirect acting carcinogens or pro-carcinogens. So here is a table showing direct acting and indirect acting. Direct acting carcinogens include alkylating agents like anti-cancer drugs, beta-propionolactone, chlorambucil. So all these were the anti-cancer drugs, epoxides. So these were the alkylating agents, and then acylating agents like acetyl, imidazole, dimethyl carbamyl chloride. So these were the direct acting carcinogens. Okay. So tumor is lymphoma, AML and bladder cancer. So these were the carcinogens. And indirect acting carcinogens include polycyclic aromatic hydrocarbons like tobacco, smoke, fossil fuel, soot, tar, mineral oils, smoked animal foods, industrial and atmospheric pollutants, and anthracenes like benza, dibenza, dimethylbenza, methylcolandrin. So these were the indirect acting carcinogens. That means they will undergo certain chemical reactions or metabolic reactions. So these cause lung cancer, skin cancer, and cancer of upper aerodigestive tract. And then aromatic amines and azodyes like beta-naphthalene, benzidine, azodyes like butterello, scarlet red, etc. So these cause bladder cancer or hepatocellular carcinoma. And naturally occurring products like aflatoxin BI, it's a fungal toxin, actinomycin D, mitomycin C, betel nuts. So these cause hepatocellular carcinoma. And miscellaneous, so nitrosamines and nitrosamides cause gastric carcinoma. Vinyl chloride monomer causes angiosarcoma of liver. So these were the carcinogens. These were the associated tumors. Okay. So direct acting carcinogens, they will directly induce carcinogenesis process, whereas indirect acting carcinogens, they undergo certain metabolic reaction, and then whatever the metabolite is there, that will act as a carcinogen. So the stages that will be taking place in chemical carcinogenesis. So direct acting carcinogens or indirect acting carcinogens, these doesn't require any metabolic activation, whereas indirect require metabolic activation. So these carcinogens, whatever form, that will directly act on this target cell. So when they get exposed to this target cell, it causes the release of certain reactive electrophiles. So reactive electrophiles were liberated. So as they are highly reactive, they will react with with the target molecules like DNA. So when these carcinogens directly react with DNA, they cause permanent DNA damage. So till here the step we call it as initiation. So the carcinogens, when they interact with this target cell and cause damage to DNA, so then we call it as till then we call it as initiation of carcinogenesis. And then so whatever the damaged DNA, whatever it is there, it undergoes clonal proliferation of altered cells. So as I said that in tumor, only a particular clone of cells has a tendency or capability to undergo replication or proliferation. So that clonal proliferation will be taking place. So that we call it as promotion. That is the second step called promotion. And so this clonal proliferation to cancer phenotype. So they'll get a certain phenotype for the physical nature, then we call it as cancer phenotype. So from that too, if it converts into malignant tumor, that means if it spreads to other parts, then we call it as malignant tumor. That comes under progression. From cancer phenotype to malignant tumor, that is progression. Okay. So target cell initiated, it may be any chemical or carcinogen. It forms initiated or mutated cells because of damage to DNA. So then with the presence of promoter, it will be converted into cancer cells, and these cancer cells will be metastasis to distant places causing the cancer, that is malignant cancer. So for the target cells to convert into initiated cell, it requires certain it follows certain steps. So the first step is metabolic activation. So the indirect acting carcinogens, whatever are there, they'll undergo certain metabolic activation in the presence of certain enzymes like P450, cytochrome P450 in the ER, that is endoplasmic reticulum, and thereby it undergoes metabolic activation. Next is reactive electrophiles that we have discussed. It will release certain electrophiles which are electron deficient, so they have a high tendency to react with other molecules. So it binds to DNA, RNA and other proteins and target molecules. So the primary target of these electrophiles is DNA. So these electrophiles reacts with DNA, causing the permanent damage to DNA. So the initiated cell is nothing but the damaged DNA which undergoes mutations, that we call it as mutagenesis. So these were the steps. Target cell, reactive electrophiles. These reactive electrophiles bind to DNA, causing damage to DNA. So this is thereby forming initiator cells. So the stimulus for proliferation may come from regeneration of surviving cells, dietary factors, and hormone-induced hyperplasia viruses. So once these initiator cells were formed, they should undergo some promotion. So they should undergo conversion to, like, they should get this cancer phenotype. So for that the promoter, the stimulus for proliferation are already regeneration of surviving cells, dietary factors, and hormone-induced hyperplasia and viruses. And next, coming to promotion of carcinogenesis. So they do not produce sudden change during this step. They require application or administration. So why? Because they require certain promoter to induce this promotion, and the change induced may be reversible from here if the dose of this carcinogen is very less, or if it is stopped and the exposure got broken down, then it doesn't cause the further progression. It may be reversible till this stage. They do not damage the DNA and are thus not mutagenic, but instead enhance the effect of direct acting carcinogens or procarcinogens. So in these promoters, they will not cause damage to DNA, but what will they do? They will support these direct acting or indirect acting carcinogens to show this carcinogenesis. And tumor promoters act by further clonal proliferation. So they are also acted by clonal proliferation and expansion of initiated cells and have reduced requirement of growth factor. So these promoters, they don't require much amount of growth factor, especially after RAS gene mutation. So after RAS gene mutation, it doesn't require much of these growth factors because continuously they'll be proliferating. Okay. So they do not produce any sudden change, these promoters, and what will they do? Till this step, it may be reversible and it does not cause damage to DNA, but it will support this procarcinogens or carcinogens, and also it will helps in the formation of initiated or mutated cells and also helps in the clonal proliferation. So these were the roles of promoters. So promoter carcinogens, their mechanism is not mutagenic. The dose, repeated dose exposure. So when the dose is continuously getting repeated, then it will promote the carcinogenesis. Response here, it is sudden response. It does not produce any sudden response. The change here is permanent and irreversible, but here it is reversible. Sequence, it is applied first, then followed by promoter. So first there will be formation of initiator genes, then followed by promoter genes, promoter carcinogens. And effectivity, it is if alone is exposed in large dose, it is not effective alone. So promoter carcinogens will cause these carcinogenesis only in the presence of initiator carcinogens, whereas the reverse is not required. Molecular changes, so here it includes mainly mutation of RAS oncogene and P53 anti-oncogene. Here it includes clonal expansion of mutated cells. So the examples for initiated carcinogens include most chemical carcinogens and radiation. For promoter carcinogens, it includes hormones and phorbol esters and also dietary changes. So the third step is progression of carcinogenesis, that is conversion of this cancer phenotype to malignant tumor. That we call it as progression. That period we call it as progression of cancer. These features pertain, so the cancer phenotype, it includes the features pertaining to morphology, biochemical composition, molecular features of malignancy. So these such phenotypic features appear only when initiated cells start to proliferate rapidly, and in this process acquires more and more mutations. So when these tumor cells will get the characteristics of malignant cells, only when it undergoes continuous proliferation. So during this continuous proliferation, so there will be continuous mutations will be there. So as a result of which these changes can be identified clearly. So this is the progression of carcinogenesis. So first, initiation, promotion and progression. So for initiation, direct acting carcinogens or indirect acting carcinogens. Direct acting doesn't require any metabolic reaction, whereas indirect require metabolic reaction. So during this process, when this undergoes metabolic reaction, it will release certain reactive electrophiles. So these will react with DNA, causing the damage to DNA. So the damaged DNA, whatever it is there, that we call it as initiated cell. Initiated cell. So this initiated cell, under the influence of certain promoters, it will undergo promotion and it will be converted into cancer phenotype. So during this, the promoters the promoters are like certain dietary factors and hormones. So all this comes under promoters, and regenerating new cells. So all this comes under promoters. So once this cancer phenotype was formed, so it undergoes progression to malignant tumor. So as it undergoes continuous proliferation, rapid proliferation. So it also gains the nature of this tumor. So that is the progression of carcinogenesis. So these were the 3 steps that were involved in chemical carcinogenesis. I think this is clear for you. And now coming to physical carcinogenesis. So it's mainly divided into 2 groups. They are like radiation and non-radiation. So in radiation, it includes 2 types of radiation, that is UV radiations, ultraviolet radiations and ionizing radiations. And coming to non-radiation, so it includes certain physical agents like trauma or any injury, mechanical injury, that comes under non-radiation physical agents. So in radiation, ultraviolet radiations and ionizing radiations. Okay. Now in radiation carcinogenesis, we'll be discussing about ultraviolet light, exposure to ultraviolet light. So the source of this ultraviolet light is sunlight, UV lamps, welders' arcs. So these UV lamps were seen in like most of the diagnostic centers. So these were seen. So humans who are exposed to these UV rays, they can cause certain form of skin cancers. But these UV radiations, they cannot penetrate so deeper, so they can reach till dermis only. So these cause a certain cell carcinoma, like dermal or skin related cancers. It includes squamous cell carcinoma, basal cell carcinoma and malignant melanoma. So these were the different types of tumors that will be seen when the patient is exposed to UV rays. And coming to mechanism, like how does this ultraviolet radiations cause carcinogenesis? By a process called induction of mutation. So this is the main or the most important is induction of mutation. So these ultraviolet radiations, they will induce mutations. So when it shows mutations, whatever the progeny or next generation cells that are coming, they were like malformed cells. So in that way, it will induce mutations and inhibition of cell division, inactivation of enzymes, and sometimes causing cell death. So out of all this, induction of mutation is the important one. And as I said that it does not penetrate too deeper, only it can reach till dermis. So that is the reason it causes skin cancers. So how does it cause this biological effect? It forms pyrimidine dimers in DNA. So UV-induced DNA damage is generally repaired. But if it is for a continuous exposure and if our body fails to repair this damaged DNA, then it causes tumors. Examples include ataxia telangiectasia in predisposed to leukemia. Bloom syndrome is predisposed to all types of cancers. Facon's anemia with increased risk of develop cancer. That means in predisposed patients, like who are having hereditary some defects to hereditary, if they are having some problems or defects to repair the damaged DNA. So in that people, it is most commonly seen. So the patients who are having Facon's anemia, they are more at risk of getting this cancer. Why? Because they have some deficiency or defect in this defect to repair the damaged DNA. So these were the predisposed patients. Why? Because they have certain genetical disorders, like in which they are unable to repair this damaged DNA. And UV radiation also induces mutated forms of oncogenes. So it will induce mutated forms of oncogenes and also mutated forms of anti-oncogenes. Mutated forms of oncogenes include RAS gene, and mutated form of like anti-oncogene is P53 gene. So these were the different genes that got affected by UV radiations. So this is the source, and it causes carcinoma and malignant melanoma. Mechanism is mainly induction of mutation, that is by it causes mutation of certain oncogenes and anti-oncogenes, especially RAS gene and P52 gene. And our body has ability to repair a damaged DNA, but the patients who are or predisposed or genetically unable to repair a damaged DNA, they will get the problems when they get exposed to UV radiations. And this can be seen even in that the patients who are working near to that regions. And coming to ionizing radiations, X-rays, alpha rays, beta rays, gamma rays, radioactive isotopes, protons, neutrons. So all this comes under ionizing radiations. So these ionizing radiations can cause cancer even in animals and also in man. Okay. Example of it includes like higher incidence of radiation dermatitis. So first, because of exposure to radiation, it causes radiation dermatitis, and even it is further getting exposed to those kind of radiations, it causes subsequent malignancy of this skin. So this is commonly seen in the workers who are working in X-rays, like normal X-ray centers or X-ray workers and radiotherapists. So in that patients you can see this. So in that workers you can see these kind of cancers, and mechanism include. So it has two possible mechanisms. First is it may directly cause any alterations in the DNA at the cellular level, and second is it may dislodge ions from water and other molecules of the cell and results in formation of highly reactive free radicals that may bring about the damage. That means these ionizing radiations, they will act upon this water H2O, and when these gets acted upon this water, they will release certain free radicals like hydroxyl radical, oxygen free radical. So these free radicals will be produced. As we know that free radicals are highly reactive, so these free radicals will react with cells DNA or other cells, causing the damage to that, causing mutations in that cells. Okay, so first is it may directly act on the DNA, or second is it will act upon water and converts this water H2O molecule into free radicals, and these free radicals will bring about this damage. Okay, so damage to the DNA resulting in mutagenesis is the most important action of ionizing radiation. So because of damage to DNA, it causes mutagenesis, which was the most important action of ionizing radiations. So it may cause chromosomal breakage, translocation, or point mutation. So when these ionizing radiations gets exposed to DNA, it causes chromosomal breakage or translocation or point mutations. So the effect of these ionizing radiation mainly depends on a radiation dose rate, and frequency. So these were the factors related to the exposure of radiation, and also it depends upon the host factors, various host factors like age of the individual exposed, individual susceptibility, immune competence, hormonal influence, and types of cells irradiated. So by considering all these factors, the damage to DNA depends upon. And then coming to non-radiation physical carcinogenesis. So if there is any mechanical injury to the tissues, such as from stones, gallbladder stones, or stones in the urinary tract, heel scars from burns and trauma, so these are at risk of causing increased risk of causing carcinoma, but there is no proper evidence supporting this phenomena. So it's maybe due to mechanical injury caused either by stones in gallbladder or urinary tract, and heel scars of certain burns or trauma. So these were the mechanical agents, but there is no exact evidence describing this non-radiation physical carcinogenesis. So with this, we have completed pathogenesis of carcinogenesis, pathogenesis of cancer. There is another small topic, that is viral carcinogenesis, that we'll be discussing in the next class. Okay. So this is all about physical carcinogenesis and chemical carcinogenesis.